All right, so let's dive into some really cool new research
Yeah.
about how the very first cells might have formed.
Okay.
We're talking way back, billions of years ago,
Right.
before off plants or animals,
Hmm.
when Earth was this like wild and chaotic place.
Yeah.
Scientists have always kind of wondered, like, how
Yeah.
did life come from all that, you know, that primordial soup?
Right.
And this new study in PNAS,
Oh, wow.
the
PNAS?
Proceedings of the National Academy of Sciences, gives us some
Oh,
exciting
yeah.
clues.
I'm excited.
Yeah.
Yeah, this is a fascinating look at protocells, which are thought to be like the precursors to living cells.
Oh, okay.
This research actually builds on like a classic experiment
Okay.
from decades ago, the Miller-Urey experiment.
Oh, right. That experiment was groundbreaking. Like, it showed that you could create some of the
Yeah.
basic building blocks of life, like amino acids,
Yeah,
by just simulating the conditions of early Earth.
right.
So how does this new research,
Okay.
take it a step further?
Yeah. In this study, researchers created like a miniature early Earth
Okay.
in a sealed reactor.
Interesting.
They filled it with gases like methane and ammonia,
Okay.
added water,
Uh-huh.
and used electrodes to simulate lightning strikes.
Wow. So they, recreated the chaotic atmosphere
Yeah,
of early Earth in a bottle.
pretty much, yeah.
So what did they find?
Well, they were expecting to see,
Yeah.
some organic molecules form,
Hmm.
similar to the Miller-Urey experiment.
Right.
But here's the surprise.
Okay.
They observed this organic film
Uh-huh.
forming on the walls of the reactor.
Oh, wow.
And even more intriguing, spherical and tubular structures hanging down from this film into the water.
Okay, now that's unexpected. Those sound like the protocells you mentioned
Exactly.
yesterday.
And to make things even more interesting,
Okay.
they discovered that hydrogen cyanide,
Mm-hmm.
or HCN, was crucial in the formation of both the film
Interesting.
and these protocell structures.
HCN, isn't that like a pretty toxic chemical?
It can be, yeah, at high concentration.
Right.
But what's fascinating is that HCN is also a key ingredient
Okay.
in the formation of more complex molecules.
Okay.
The researchers found that the organic film only formed in the presence of water,
Right.
and water is essential for HCN molecules to link up
Okay.
and create larger chains called polymers.
Got it.
Think of it like Legos,
Yeah.
where each HCN molecule is a single block,
Okay.
and water acts as the connector, allowing them to snap together.
Oh, that makes sense. So water is the key
Yeah.
to building these bigger molecules.
It is.
But you mentioned something else playing a role, right? Silica?
Yes. The reactor was made of borosilicate glass,
Okay.
and the silica in the glass actually speeds up
Oh, wow.
the synthesis of those HCN polymers.
Interesting.
It acts like a catalyst,
Uh-huh.
facilitating the reactions without being consumed.
So it's like silica's turbocharging the whole process.
Exactly.
And how do those bubbles we talked about earlier fit into all this?
Okay, well, as all these reactions are happening,
Yeah.
they produce gases,
Uh-huh.
which build up beneath the organic film, forming bubbles.
Okay.
And these bubbles are filled with the precursors to those HCN polymers.
So these aren't just any ordinary bubbles. They're packed with the ingredients for protocells.
Yes.
What happens next?
When the bubbles come into contact with the water,
Mm-hmm.
the HCN precursors solidify
Okay.
and form a shell around the bubble.
Interesting.
It's like the bubble acts as a mold,
Uh-huh.
and the HCN material uses it to create a three-dimensional structure.
Wow, that's incredible. So basically, these bubbles, along with silica and water,
Yeah.
are creating these potential precursors to life. What do these protocells actually look like?
The researchers observed both spherical
Mm-hmm.
and tubular structures,
Okay.
and it's thought that the shape might actually reflect different stages of development.
Wow.
The tubular ones even seem capable of dividing,
Really?
similar to how bacteria reproduce.
So even in this non-living system, we're seeing hints of processes that
Wow.
we usually associate with life.
Exactly.
And the fact that these protocells are hollow is significant too.
Absolutely. That hollow space could have acted as a protected environment.
Oh.
A tiny container where those crucial molecules needed for life could concentrate and interact.
Like little tiny test tubes.
Exactly like microscopic test tubes where the very first steps towards life
Wow.
might have been taking place.
It's mind-boggling to think that something as simple as bubbles
I know, right?
could play such a pivotal role in the formation of life as we know it.
It really challenges our assumptions
Yeah.
about the complexity of life's origins.
Sure.
And it raises a fascinating question.
Yeah.
If these protocells could arise abiotically,
Mm-hmm.
meaning without life being involved,
Right.
What does that mean for our search for life
Yeah, that's a great question.
elsewhere in the universe?
Before we jump into that,
Yeah.
I think we should let our listener take a moment to digest all this incredible information.
It is a lot to take in.
We'll be back in a flash to explore those implications and delve deeper into the fascinating world of protocells.
Sounds good.
Yeah.
Welcome back.
Yeah, those protocells forming around those bubbles. It
Yeah.
reminds me of something I was reading about how scientists are using
Right.
those microfluidic devices to,
Okay.
create artificial cells.
They're using tiny channels
Yeah.
to control the flow of fluids and create these synthetic compartments
Yeah.
that mimic the structure of cells.
It's like they're taking inspiration from,
Wow.
nature's own design principles.
Exactly. And it just highlights how interconnected science is.
Yeah.
Like we're learning from the past to create the future.
Yeah.
In this case, the ancient process of protocell formation
Right.
might actually inform, the development of new technologies.
Yeah, it makes you really appreciate the ingenuity and adaptability of life,
Yeah.
both on Earth and potentially elsewhere.
Yeah. And speaking of elsewhere, let's get back to that question about the implications of this research
Yeah.
for the search for extraterrestrial life.
Right. So if these protocell structures can arise through purely abiotic processes,
Right.
it means we need to be extra cautious when interpreting potential biosignatures on other planets.
You mean the signs that might indicate the presence of life?
Yeah, past or present.
Precisely.
Okay.
Before this research,
Yeah.
finding fossilized evidence of those bubble-like structures on Mars
Yeah.
seemed like a slam dunk for ancient life.
It would have been so exciting.
But now we have to consider the possibility that those structures
Yeah.
might not be biological in origin.
Yeah.
They could be a product of this abiotic process
Yeah.
we've been discussing.
It adds a new layer of complexity to the search for extraterrestrial life.
It does.
Astrobiologists are going to need to develop even more sophisticated techniques
Yeah.
to distinguish between true signs of life
Mhm.
and these fascinating but ultimately non-living protocells.
Yeah, it's a challenge,
Yeah.
but it's
Yeah.
also what makes this field so captivating.
Right. And while it might complicate things in some ways,
Yeah,
it also opens up a whole new realm of possibilities
it does.
for where life might exist.
Instead of looking for planets that are perfect replicas of Earth,
Right.
we can broaden our search to include environments that might seem
Yeah.
less hospitable at first glance.
Exactly. Instead of looking for Earth 2.0,
Right.
we can start thinking more broadly.
Yeah, exactly. It expands the possibilities for finding life as we don't even know it.
Right.
We might be surprised by the diversity of life forms
Yeah.
that could exist out there.
It's like the universe is telling us, don't limit your thinking.
Exactly.
And speaking of not limiting our thinking,
Okay.
let's zoom back in on this research.
Okay.
There's something we haven't talked about yet.
Okay. What's that?
The role of silica after the protocells have formed.
Oh, you mean how it's not just involved in the formation process,
Yeah.
but might actually become
Yeah.
part of the protocell structure itself?
That's right. The researchers found evidence
They did.
that silica might be incorporated into the walls of the protocells.
Interesting.
And that got me thinking about
Diatoms?
diatoms,
Yeah.
those microscopic algae
Yeah?
with those beautiful silica shells.
Yes.
What made you think of them?
Well, it's like nature found a way to use silica
I see.
to build these intricate structures,
Yeah.
both in these ancient protocells
Yeah.
and in modern day organisms.
Right.
It makes you wonder about the potential for silica to play a role
Yeah.
in the evolution of early life.
That's a really interesting connection.
Yeah,
It
it is.
highlights the continuity of life, how certain elements and principles
Yeah.
keep recurring throughout evolutionary history.
Yeah, and it makes you wonder if silica might be a key ingredient
It could be.
in the development of life
Yeah.
elsewhere in the universe as well.
That's a cool thought. It's amazing to think about how these seemingly simple building blocks,
Yep.
bubbles, silica water, and HCN,
Right.
could potentially give rise to something as complex and extraordinary as life.
It really speaks to the power of emergent properties.
Right.
How complex systems can arise from the interaction of
Right.
simpler components.
And the researchers made another fascinating observation.
Oh, yeah.
They notice that the thinner ends of the protocells,
Right.
especially those tubular ones, were always connected to a tiny opening in the organic film.
Oh, interesting.
almost like a little doorway.
Ah, yes.
Yeah.
That suggests a potential pathway for communication with the surrounding environment.
Exactly. It means that the protocells weren't completely isolated.
Right.
They had a way to exchange materials and energy with their surroundings.
Right, which is crucial for life.
Yeah, which is crucial for life as we know it.
Think of it like a city.
Okay.
With those openings acting as gates or channels, allowing nutrients to flow in and waste products to flow out.
Right. It creates like a dynamic system.
It does.
A network of interactions that might have been essential for the evolution of those early protocells.
Yeah, it's almost like they were plugged into a network, sharing information and resources with each other.
That interconnectedness is really at the heart of life.
It is.
And it's not limited to biological systems.
Wow.
Even in the abiotic world, we see this web of interactions and feedback loops that drive complexity and change.
It makes you realize that life might not be this isolated phenomenon, but rather an emergent property of complex systems.
Right, arising from the interplay between simple building blocks and their environment.
It's a profound thought, isn't it?
It is.
And it reinforces the idea that life might be more common than we think, woven into the fabric of the universe, in ways we're only beginning to understand.
I'm still reeling from all the amazing insights we've uncovered today.
Yeah, me too. It's a lot.
And I'm sure our listener is too. What stands out to you? The most significant takeaway?
For me, it's the realization
that
the journey from non-living matter to life might be more subtle and intricate than we ever imagined.
Right.
It's not necessarily like a giant leap, but rather a series of small interconnected steps. Each building upon the last. And those steps might be happening all around us, even now, in ways we're only just beginning to comprehend.
I love that perspective. It highlights like the beauty and wonder of the scientific process. How each discovery opens up new avenues for exploration and pushes us to reevaluate what we thought we knew.
Exactly. And it reminds us that we're still just scratching the surface. There's so much more to discover about the origins of life and the vast possibilities that exist in the universe.
Before we wrap up this incredible deep dive, I want to encourage you, our listener, to keep exploring, to keep asking questions. And to never stop marveling at the incredible mysteries.
It's an amazing universe out there
that surround us.
And if you have any thoughts or questions about the research we discussed today, we'd love to hear from you. You can find us on social media or visit our website to join the conversation.
Until next time, keep diving deep. You know, it's amazing to think we started with bubbles and silica. And now we're talking about like the interconnectedness of life and the vast possibilities in the universe.
It's really cool how like even the simplest things can lead to these like profound insights. This research is definitely like made me reconsider what it takes for life to arise.
Me too. It's almost like the universe is full of these like hidden pathways to complexity just waiting to be discovered.
And it makes you wonder what other seemingly mundane things might have played a role in the emergence of life.
That's a question that keeps scientists up at night.
Oh, yeah, for sure.
I guess that's what makes this research so exciting. It's not just about answering questions, but also about opening up new ones.
Exactly. And this particular study raises so many fascinating avenues for future research.
Like what?
For instance, we still don't know the exact chemical reactions happening inside those protocells.
Right. We know that those HCN polymers are interacting with like water, silica, and other molecules, but how are they actually forming the building blocks of life? What are the specific steps in that process?
Those are like key questions that scientists are eager to explore.
For sure.
And beyond that, we need to investigate other potential pathways to life.
Right.
This research focused on specific conditions.
Yeah.
But what other chemical combinations and environments might be conducive to life's emergence?
The possibilities are endless.
I know, it's so exciting.
And with every new discovery, we're getting a clearer picture of the incredible diversity that might exist out there.
It's truly humbling to think about.
It is.
And as we continue to explore, both here on Earth and beyond, we need to refine our techniques for detecting signs of life, as we've discussed. We need to be able to distinguish between true biosignatures and those that might arise from abiotic processes.
It's a challenge, but one that I think scientists are ready to tackle.
Oh, yeah, definitely.
After all the search for life beyond Earth is one of the most fundamental questions we can ask.
I completely agree. And this research gives us hope that the answer might be closer than we think.
Oh, wow.
If life can emerge from something as simple as bubbles and silica, then perhaps it's not so rare after all.
That's a beautiful and profound thought.
It is.
And as we wrap up this deep dive, I hope our listener is left with a sense of wonder and a thirst for more knowledge.
We encourage you to continue exploring, to keep asking questions, and to never stop marveling at the incredible mysteries that surround us.
It's been an incredible journey.
It has.
Exploring the world of protocells and the origins of life.
Absolutely.
Until next time, keep diving deep.